So like I said, if you have any questions about the midterm, you can talk to me, and please take a chance to review it and make sure you understand the answers. Midterm 2 does not cover the same topics. Midterm 2 covers topics 7 to 12, so we're not going to revisit that material in midterm 2. So I want to talk about the lab 3, a little bit about the discussion. So the discussion is worth, I think it's 30% of the full report. So basically 1/3 of everything you're doing for this formal report. You're looking at probably around 3-ish, maybe a little bit more pages of written material. And this is where you're going to discuss everything. So the results section we talked about last day, where you're just presenting the results. Here are the results, here are the graphs. You know, you're gonna mention the trends that you see. The discussion is where you're going to interpret those results. So you're going to do a little bit of rehashing where you're talking about the trends, and then you're going to kind of explain those trends. Why did you see what you saw at -20 degrees? And why did you see what you saw at 85 degrees? Why was the propanol so different than the water, and so on. And there's questions in the lab manual that kind of guide you through all the things you should discuss. So make sure you do go through those questions in the lab manual and that you do answer every single one of them as part of the discussion, because that's what I'm going to be looking for when I grade it. So as always, you know, be thorough, be specific, be complete. This is your chance to show me that you understand what you're talking about. I'm not just scanning for keywords, I'm reading everything. So do make sure that you are using proper grammar and proper language and all those kind of things. So I have an example here, as usual, of some writing. And this one here, it's probably obvious, again, right away, which one is better. You know, the first sentence there, clearly temperatures below freezing are damaging to bead membranes. Hey, that's not bad, but that's a start. It's not specific. It's not, you need to flesh that out. You can see the second sentence I have there is more specific. It's again not everything, it's just a start. You can see already I'm being specific, -20 degrees, right? That's specific. It's actually telling me exactly how much damage, right? We have 16 micromolars of beta-cyanide leaked. So that's very specific. It's giving me an idea when I read it. It's not vague. It tells me, okay, -20 is bad for these membranes. Additionally, you can see it's referring to the figure. So that way, if you know, me as the reader, I can read it and say, oh well, okay, I want to see what's going on at -20. I can go back to that figure and take a look at your data. So like I said, both of these are kind of a start of that discussion. Also in your discussion, like I said, you're going to attempt to explain why you're seeing what you're seeing. So you can see there's another sentence, temperatures below freezing are damaging because, you're going to look for some answers on that and try to explain what you think is going on. And, you know, it doesn't hurt to spend a little bit of time on Googling these kind of things, or, you know, taking a look at your class notes or textbook and seeing what you can find around membranes and temperatures, because you're going to find some answers there. Probably people who've thought about this as well before. At some point in your report, I think I mentioned this before, you're going to either do this in your introduction or you're going to do this in your discussion. I think more people end up doing this in their discussion, is you're going to cite something. You're going to cite a scientific paper. Now, if this was a third-year course or something like that, you would have dozens of citations. And citations, what they do is they add authority to what you're saying. Okay, if you're telling me that membranes are damaged because of this, that's great. Maybe you're 100% accurate. But if you're citing something as well, then you're showing that it's not just your opinion, but there's research on this stuff. And so it's always a good idea to cite things. Like I said, as you go on in your studies, it's important you cite a lot of things, because that's an academic way to do things. At this level, I'm looking for one citation, and doesn't really matter too much how relevant it is. I'm looking to see that you actually cite properly and do proper referencing format. That's what I'm looking at. I'm going to talk about citations and referencing a little bit on Wednesday. As for today, I want to move on from all this stuff. So keep in mind, if you have any questions about the reports, you want to show me a rough draft or anything like that, I'm available before Thursday to do so. So one last thing about the report, I think I showed you this exact slide before. I'm looking at probably most people are going to have about 10 pages, you know, give or take a little bit. And in terms of the length of things, this is what we're looking at, right? So just a reminder of that. Hopefully everyone has worked on it at least a little bit, and you're kind of getting to the point now where you're polishing up sections and eventually going to put them all together. And your report is going to be beautiful. All right, so moving on. Okay, I guess we did that already. We took up the midterm. And so we're going to go back to topic 6. There were just a few things to finish off on topic 6. So we had talked about eukaryotes in topic 6, and we talked about the endomembrane system, although we didn't quite get to vacuoles and peroxisomes. So unfortunately got a little bit behind. So I'm going to talk about them now. And mitochondria and chloroplasts shouldn't take us too long. And some of those things may end up on the next midterm. Definitely mitochondria and chloroplasts will, because we're going to talk about respiration and photosynthesis. So let's talk about lysosomes. So lysosomes, that first part of the word lys means breaking. So they're going to break things. And zome usually means a body or organelle or something like that. So these are like cellular stomachs, basically. In fact, they're very much like stomachs in that they are low pH environments and they're used for digesting things. So you can see in this particular diagram here, we've got some food particles here, and they're being brought in by endocytosis, and they form a food vacuole right here. And that food vacuole eventually fuses with one of these lysosomes, and the lysosome releases hydrolytic enzymes into the food vacuole, and it all gets digested. So in this case here, I'm not sure why. Sometimes this happens that some waste products here are expelled by exocytosis, but lysosomes are of course involved in that. Notice these lysosomes are coming from the Golgi body. So the proteins, the digestive proteins would have been made up here in the ER, eventually goes to the Golgi, and then they become these little secreted vesicles, and their pH gets lowered and they become a lysosome. And that's how they work. Here's some more pictures of some lysosomes. There's that word phagocytosis. We talked about that already. And this means cellular eating. And you can see the lysosome is digesting some sort of food particle. Another process that's worth knowing, at least the definition for, is something called autophagy. So auto means self and phagy means eating. So this means basically the destruction of cellular parts. And so you can see in this diagram, it looks like there's an old peroxisome and an old mitochondria. Sometimes these things, you know, they've outlived their functionality, and you can't have these organelles that are loaded with enzymes floating around and doing damage. So this autophagy process, a lysosome goes around and digests these things, and they're no longer dangerous. So that's what lysosome is. I know I had a couple questions about lysosomes in the sample midterms, and probably some people were wondering what that was all about. So now you know. So what about vacuoles? Turns out we've actually talked about all 3 of these types of vacuoles already. We have food vacuoles. We just talked about those a moment ago. They are made by phagocytosis. We talked about central vacuoles when we talked about lab 3, and you can see in this case that here's a central vacuole here in this plant cell. The membrane that surrounds the central vacuole is the tonoplast, and what's in that central vacuole? Mostly water, and depending on the plant type or the cell type, there may be other things. So in the beetroot, we know that there's a pigment in there, the beta-cyanin pigment, which is kind of a purpley pinkish color. In other parts of the plant, you might have sap in there as well, but it's mostly water. And it's basically a massive storage compartment in the middle of a plant cell. And that's what it's doing. I think I have another picture here of a central vacuole in a plant. They're most notable in that, and then they take up a huge portion of the plant volume, usually even bigger than the nucleus. And then the last type of vacuole that we talked about was the contractile vacuole. And we were talking about them in the context of paramecium. And so that's going to come up in this week's lab. If you watch that video there, right where the arrow is, if you watch it carefully, every once in a while you're going to see that contractile vacuole. It's like this circle that sort of shrinks, and that's used to pump out excess water. So these are organisms that do not have cell walls, and they live in ponds, which is a hypotonic environment. And in that pond, you're going to end up with water rushing into the cell all the time. So they pump out the excess water. That's what a contractile vacuole is. So peroxisomes, the last one on there of the endomembrane system we haven't talked about. I'm not going to get into a lot of detail on peroxisomes. We kind of missed our chance to do that. But they do come up. They're organelles, and they usually have a lot of specialized functions. You can see it's mentioned there that these functions include oxidation reactions. So to give you an idea of some of the things that are going on, if you're an animal cell, sometimes you eat food and it has fatty acids that are weird. They're like extra long, or they're branched or something like that. And normally normal fatty acids are actually broken down in the mitochondria. But the fancy ones, the mitochondria can't break it down. So they go to a peroxisome, and oxidation means breaking down. And often in these breakdown reactions, hydrogen peroxide is produced. So hydrogen peroxide is this one thing here, H2O2, and that's a really reactive compound. And so peroxisomes will actually not just make hydrogen peroxide, but they will also break it down. So kind of the bottom line to know is that these have specialized oxidative functions, and often involves hydrogen peroxide. And I'm not really going to ask you much more about them. I'll show you one more type of peroxisome that is used in plant seeds. It's even so specialized it has its own name. It's called a glyoxysome. And so you may know what a seed is. A seed is a baby plant, not even a baby plant. It's a pre-plant, right? It isn't a plant yet, and it has a lot of fat in there. And that fat, when the germination process occurs, gets turned into sugars. And that's what the peroxisomes do. They convert the fatty acids to sugars as a part of that germination process. So like I said, peroxisomes, lots of specialized functions. I'm not going to come back to them at all this semester. Okay, last two organelles to talk about are mitochondria and chloroplasts. And I'm also not going to talk about them a lot now, because I am going to talk about them much more when we get to respiration and photosynthesis. But a couple of things that need to be said about them. First thing is that they are not part of the endomembrane system. So what does that mean? It means they're not connected to the ER or the Golgi in any way that all those other organelles are. So they're not connected directly, and they don't have vesicles from the other organelles coming and fusing with them. So they kind of have some independent ways to do things, these mitochondria chloroplasts. Notice that they have their own DNA and ribosomes, So some of their proteins are actually made in-house. So if you think about the human genome has a bunch of DNA in the nucleus and a little bit of DNA in their mitochondria. We don't have chloroplasts. Hopefully you knew that. So these are unique organelles involved in respiration and photosynthesis. Here are some mitochondria. They're almost always colored red or orange in textbooks because, of course, they are oxidizing fuels, and oxygen is often depicted as red, but they're not red. That's just a color that, for some reason, textbooks and artists have associated with mitochondria. So mitochondria is a plural word, by the way. Mitochondrium is one. And like I said, I'm going to come back to these a little bit later when we talk about respiration. But notice it has two membranes, an inner membrane and an outer membrane. The innermost part of this is called the matrix. And the inner membrane, it kind of makes all these folds that kind of look like little fingers. So I'm talking about these little things here. And sometimes those are called cristae, and it just means the inner membrane. And that's where the electron transport chain occurs. That's where a lot of the chemical reactions of the mitochondria occur. So here's another picture of mitochondria. I showed you one of these before, kind of as the cartoon blended with the the electron micrograph, and often they are kind of bean-shaped, I guess you could say. Here's another one, a cross-section of TEM, and you can see all the inner membranes there. So what do mitochondria do? They're involved in cellular respiration and a few other cellular processes. So we'll talk about them more in a couple of units from now. There's also the chloroplasts. Sometimes chloroplasts are called plastids. I'll explain that a bit more when we get to photosynthesis. And this is where, of course, photosynthesis occurs. And so if you look at this diagram of a chloroplast, it's kind of like a mitochondria. It has extra membranes. In fact, it has 3 membrane systems. It's got an inner membrane, an outer membrane, and then this innermost thing here is called a thylakoid membrane. And that is green. That is the green part of the chloroplast. That's where the chlorophyll is found in those things called thylakoids. So more on thylakoids when we talk about photosynthesis. And so that's a little bit later on. Here's a photograph of some of the chloroplasts that we were looking at in, what was that, lab 2, I think it was, in the aquatic plant Elodea. And Elodea should be underlined or italicized. So there's the underline. There's some more pictures. Sometimes these chloroplasts and actually mitochondria aren't always necessarily bean-shaped. Sometimes they're all connected in these big networks, which is kind of an interesting thing that we've discovered in the last few years. So I was just showing you this last picture of a chloroplast because I just want to highlight, of course, that there is starch in a chloroplast. And why is that? Because that's where it's made. And sometimes in some plants, they actually store their starch and or glucose or other products there as well. So one last thing to talk about mitochondria and chloroplasts is this whole thing about, hey, they're not part of the endomembrane system, and they have their own DNA and they have their own ribosomes. And as a consequence, this theory has been floating around for a long time now, in that the thought is that mitochondria and chloroplasts, they're not like the other organelles, and it's believed that maybe they had a different origin. And what origin would that be? It's believed that mitochondria and chloroplasts were maybe some sort of ancestral, some sort of maybe bacterium. And you can see this is how the hypothesis works. You've got some sort of eukaryotic cell, and it's bringing this aerobic bacterium in by endocytosis and doesn't kill it. Instead, they start to live together, and that bacterium eventually has become a mitochondria. And same thing with the chloroplasts, that they were some sort of cyanobacteria, and that they were brought in by endocytosis and learned to live with that cell. So it's not the perfect theory, but there is some decent evidence to suggest that something like this is true, or at least that there's some good evidence to suggest that this theory has some good merit. And I was going to just write down some notes here now. So we'll call this supporting evidence for the endosymbiotic theory. Supporting evidence, and I mentioned some of these things already. One thing I'm not going to write down, because it's just almost too obvious, is that bacteria are kind of bean-shaped, and so are mitochondria and chloroplasts, and similar sizes. So you can write that one down, but I'm not going to write that one down because I think that one is just it's okay, but it's not as important as the other evidence. So I'm going to, by the way, abbreviate mitochondria as MT and chloroplasts as CL. Okay, so I don't have to write the whole thing out. Okay, mitochondria and chloroplasts, they have their own DNA and ribosomes. Okay, so that's a good point. So that DNA is circular. And where have we seen, I can't spell here today, or can't write. The DNA is circular. Where have we seen circular DNA before? We have seen circular DNA in bacteria. So that is kind of something similar. The ribosomes. The ribosomes are 70S. So where have we seen 70S ribosomes before? We've seen those in bacteria. They have an extra membrane. Why is that important? Because most organelles only have one membrane. These guys have at least one extra. In the case of chloroplasts, they have two. And why is that important? Because this suggests phagocytosis, which suggests phagocytosis. So that's kind of, some of the main evidence that suggests this. The last thing I'm going to put, it's just an extra bullet point, is if you look at genetic sequences. This is an area that I'm not as familiar with in terms of, what's going on. But if you look at the genetic sequences of, genes that are found in mitochondria and chloroplasts, there's a lot of similarities to similar genes found in bacteria and cyanobacteria. And so, that suggests a common origin anyway. So anyway, endosymbiotic theory, very interesting, and makes these organelles certainly a little bit more fascinating than some of the other ones. And so make sure you know some of those things for the next midterm, because there'll probably be some sort of question about this on there. Okay, so this is where I wanted to be today. So I'm hoping, I'm thinking we're in good shape though, that we won't be behind for too long. But we're going to talk about cytoskeletons today, and at least start talking about cytoskeletons today. And we'll finish this topic on Wednesday, of course. So we did talk about the cytoskeleton a little bit already, and I've given you some examples a little bit here and there already. And I mentioned I'd come back to it. So the cytoskeleton is this network of protein fibers that are found throughout cells. And you can see from this image here that they're quite extensive, and there's also different types. I'll show you some other pictures of some cytoskeletons. There's another one there. You can see some pretty stunning colors. You can see the nucleus there is nice, looks like a big eyeball. So what is the cytoskeleton? You can see it says here it's a network of proteinaceous fibers. So what does proteinaceous mean? It means it's made out of protein. And they extend throughout the cytoplasm, but also other parts of the cell, like in the nucleus, you can find cytoskeletal components. This is a little cartoon showing the three different types. We have microfilaments, intermediate filaments, and microtubules. And we're going to talk about them a little bit in this week's lab as well. So by the way, that is the worst thing about the cytoskeleton, is these names. Microtubules, microfilaments, intermediate filaments. If you've never heard of these things before, they all kind of just sound the same. So I apologize for that. I didn't get to name them. But there are some things about the names that are hopefully helpful. Microtubules, for example, are actual tubes. So maybe that will help you picture and envision these things in your head. So we're going to talk about all three, not all three today. We're going to cover a little bit of microtubules today. And, oh, you know what? I just remembered. I really got to apologize for this. I meant to go back to this table and fill in for those last few organelles. So sorry about that. Why don't I do that, and then we'll introduce microtubules. That will probably be the end of today. So here are the things that we just talked about. Lysosomes, vacuoles, peroxisomes, and mitochondria and chloroplasts. Okay, so we'll throw in some notes here. Lysosomes are digestive compartments. And we'll say they're used in phagocytosis and autophagy. There we go. Vacuoles, so three types to know. We had, what were they? We had food vacuoles, and these were formed by phagocytosis. We had contractile vacuoles. And these pump water out. And what was the last kind? We had the central vacuole in plants. These are in plants, and there's some found in a lot of fungal cells as well. So I'll put fungi in there as well. So central vacuole, what's its function? Storage of water and solutes. So like I said, it depends on the part of the plant what those solutes might be. It could be sap, could be a pigment, may not be much more than just water. Okay, peroxisomes. These are site of oxidative reactions. And the example I gave you was breakdown of unusual fatty acids and branched or long ones. Mitochondria is the site of cellular respiration. And I did tell you one other thing, oxidation of usual fats. Mitochondria can break down all sorts of different types of food. We're just usually used to talking about glucose because that's what we learn about in school, but we do not just eat glucose. We do eat other things. So mitochondria is involved in those things. And chloroplasts, hopefully this one is very obvious, site of photosynthesis. Okay, sorry about that I forgot to do this table. I think it was important to kind of finish off those notes, make sure the table is complete. So like I said, if you ever want these notes, just send me an email and I will forward them to you. It's not really a big deal. Okay, so back to the PowerPoint. I want to talk about the cytoskeleton, and we're going to talk about microtubules. So there's the microtubules, and they are the largest, and they're like tubes. There's the microfilaments. These are the smallest, and they're kind of like a rope. And I'll re-review these next lecture, by the way. Like I said, I know the language on these things gets confusing because the names sound so similar. And the intermediate filaments, well, they are intermediate. They're kind of medium in size. So if you take a look at these exact sizes, we're talking about 25 nanometers here, 7 nanometers here, 8 to 12 nanometers, which is intermediate for the intermediate filaments. So one thing that you may notice in a lot of the diagrams that you see in textbooks, particularly when they're using fluorescence microscopy, is that microtubules are often stained green in fluorescence. Intermediate filaments blue and microfilaments in orange. And this is actually just has to do with the type of stains that they're using. And so this is pretty common to see these microtubules in green, for example. You may notice from this diagram too that these different types of cytoskeletal structures are actually found in different parts of the cell. And so you can see that when we talk about the functions of these, that the microtubules, for example, are deep into the cytoplasm, kind of in the middle of it, whereas the microfilaments are kind of closer to the membrane. And so anything that's having to do something to do with the membrane is going to have something to do with the microfilaments for sure. And the intermediate filaments, they're usually a little bit more specialized, and some of them are found in the nucleus. So all of these have different protein subunits. I'll come back and review this next day. I know that in your notes, I think on the first page of this, it mentions that you might want to make a table, and I will fill out that table as we go along, but probably mostly, I'm not even going to look at that today. What I will do is I will do that on Wednesday. So let's talk about these microtubules. So like I said, microtubules are actually these big tubes. Nothing is going through the tube, by the way, and I'm not sure why it forms a tube. I think it's just the way that these monomers get together in a polymer. And it's made out of these little things called tubulin. There's actually two types. There's alpha tubulin and beta tubulin. Actually, there's a gamma tubulin as well, but you don't see it as often. And you can see they're involved in a whole bunch of different processes. So you can see I'm going to talk about most of these processes here are important functions of microtubules. here are the microfilaments, so I'm just going to come back to them next day, intermediate filaments as well. So let's take a look at the microtubules. I actually have that little box in the corner is a short 30-second video, so I'm going to play that for you right now, and you're going to see the microtubules form the big fiber. So here goes.
A microtubule grows by adding tubulin units that randomly bump the growing end. We're showing only those dimers that join the microtubule. Microtubules shorten by losing tubulin. Actually, microtubules usually grow and shorten at just one end.
So that's it. Told you it was a short video. There's lots of really long ones I found on YouTube. That was a nice short and sweet one. Sometimes short and sweet is better. So we have heard of microtubules before. Sometimes they go by another name. They go by the name of spindle fibers. And so I'm just going to talk about spindle fibers briefly here. And like I said, I'll come back next day and maybe I'll spend a little bit of time defining these things a little bit better. So you probably have heard of centrioles or the centrosome. And if you take a look carefully at this diagram, these are actually microtubules. Each of these, this is like a triplet, 3 microtubules together. So you can see it's like one tube like that, and there's a second one beside it like that, and then the third one is over here. So those are microtubules, and the centrioles or centrosomes, and I'll kind of, like I said, I'll come back and define these a bit better next day when we have time. But this is kind of like the origin, the starting point of microtubules. So they grow out of the centrosomes and form these things that you probably know as spindle fibers. And spindle fibers are, of course, involved in mitosis and meiosis, and they're microtubules. That's just sort of the special name we use for them in mitosis and meiosis. So you can call them spindle fibers, or you can call them microtubules. So you can see on the left there we have an anaphase cell, and that cell is dragging its sister chromatids to both of the poles of the cell. On the right we can see the centrosomes or centrioles. So by the way, 2 centrioles makes a centrosome, right? So like I said, I'm going to come back and define these next day. I know there's centrosomes, centrioles, and centromeres, and all those words are easy to kind of get mixed up. So we'll come back and do that when we have a little bit more time. I'm just going to show you a couple more pictures of microtubules, and this is a video, and then we'll wrap up for today. So I wanted to show you this video because this is actually 3 cells. Maybe you can see we've got, for example, there's a nucleus right there. And this is in real time. And I just wanted to show you because these microtubules, they're not just these structures that sit around and do nothing. They're actually constantly growing and moving things. And you can see that here in this particular diagram. We've got, they're moving around the cell, and they're moving, in this case, probably vesicles are moving around the cell from these microtubules. Yeah, I'm not going to go there, but maybe I'll just mention they are part of flagella, and we're going to talk about flagella next day. So that's actually a good place to stop. Anyway, so thanks for coming out today. I will see you on Wednesday. Make sure you drop me a line if you have any questions about the midterm or any questions about the lab report, and I'll see what I can do to help you out. Other than that, I hope you had a good break and you have a wonderful evening.